right. Hi, everyone. Welcome. Thank you for coming to our Science to Go presentation. While we're waiting for other participants, I'm going to share my screen and show you some of the materials that you have in your science kits that you can get ready for our activities today. And it may help you in your classroom or at home to separate out the materials for the two different activities we're going to be doing. One will be a mushroom gill dissection, and another will be for a yeast experiment. So you can use the images on your screen to help separate out those items. So for the gill dissection, we need our little baggie with fungus and our tweezers. We need our napkin, an index card, and eventually we're going to need some warm water. For the yeast experiment, we'll need our sugar, our yeast, a bottle, and your bottle may be round or square, a balloon, and eventually we'll also need some warm water for that as well. Okay, while we're waiting, go ahead and get your materials out. And if you have any questions, please feel free to put those into the chat and let me know. And we'll give just a minute more for anyone else that is coming in. And captioning is available. Instructions for how to get Spanish language interpretation or captioning will be in the chat. Right, and if you're just coming in, we have some instructions on the screen about materials to separate out to get ready for our activities. So thank you for joining us. We're excited to have you here for our program. Okay, I'm going to go ahead and start our program, and anyone else that wants to join us can join as we go along. So I'm going to go ahead and start and introduce myself. My name is Karen. I'm the head of education at the Smithsonian Environmental Research Center, which is called CERC. It's a lot easier to say. It's located in Edgewater, Maryland, which is south of Annapolis. Now, we're going to take you on an adventure today with fungus, and we're only going to take questions at the end of the program for the scientist. But if you have questions in the meantime, you can still put them in the chat and we'll try to get those answered. So you can put them in the Q&A or in the chat, and we'll try to make sure that all of your questions get answered at the end. So, and like I said, there is interpretation available. Information for how to access that is in the chat. So to start with, I want you to think in your head, put on your thinking caps, because we're going to go on a journey with fungus. What is fungus? Have you ever seen it before? Maybe somewhere in a grocery store or something that you've seen in a forest, on a walk. Have you ever seen it? And what is it? Is it a plant? Is it an animal? What type of thing is it? So go ahead and put that in the chat if you have an answer. So while folks are typing that in the chat, I want to introduce you to today's theme. Our theme is fungus is our friend, because fungus is found in our forests and it's found in our food. And that's what we're going to explore today by meeting two of our scientists, Dr. Melissa and Dr. Aranga, who study fungus here at CERC in the Smithsonian. And they study the relationship between fungus, trees, and plants. Now, before we get to our researchers, we need to actually get our experiments running so that while we talk, they're going, all right? So the first thing I want you to do is push up your sleeves, because we're going to be dealing with a little bit of water. We want to make sure that you don't get wet. I want you to make sure you have all the supplies from your kits. And we're going to turn on our document camera so we can show you how to put together all of our materials. So our hand model, Riley, has all of our materials ready to go. So the very first thing we need to set up before we can do all our experiments is our fungus. All right, so we're going to start by getting out your little baggie. In that baggie, you should have some tweezers and some dehydrated or dried mushroom gills. And then what we're going to do is we're going to leave those mushroom gills in the baggie, and you're going to fill it about halfway full with warm water. Now, your teacher may have warm water in the classroom with you, or you may need to go to the sink. But we need to have that warm water so that we can rehydrate or activate the fungus to inflate so we can see it better and make some observations about it in just a little bit. Now, while our hand model is filling that up with water, a couple of things. It's important that you seal the bag up very well. And when you're done sealing it up, just put it to the side with the other materials we'll need in just a little bit to make our observations. And this includes our napkin and our index card, as well as our tweezers. Now, it's important, please don't pound on the bag or hit it with a pencil or poke it with your tweezers, because if all that water runs out, you're not going to be able to make good observations later on. Okay, so we're going to let that sit for just a minute, and we're going to go ahead and set up our other activity so that it can run. So what you want to do is you want to take your bottle. So that bottle could be rounded, it could be squared off, but we want to take our bottle and we want to fill it with our yeast and our sugar. And after that, we're actually going to put some water in. But first, let's see our hand model. We're going to open the yeast. You might want to use scissors, but you might also be able to tear it. But we want to put in some yeast. Now, yeast is just a dehydrated or dried form of fungus, just like the dried mushroom gills that you saw earlier. And it's in the same family as things like the mold on cheese and the fungus between your toes, but this is yeast. And so yeast is used in making bread and other foods. It's very important. Now, because the yeast is asleep, we need to wake it up and give it a little bit of food. So the sugar is the next thing that you're going to pour into the bottle. So go ahead and open your sugar packet up and pour that into the bottle. And once you've poured the sugar into the bottle, you might want to take a smell very cautiously over the top of the bottle. What does that yeast smell like? Can you smell anything? What do you notice? In just a little bit, when the experiment's over, we're also going to smell the yeast again and see if it smells differently. All right. Now, to wake the yeast up, we also need some more warm water. Now, for your bottles, what you want to do is take 3 fingers and lay them against the bottom of the bottle and fill up the bottle to the top of that finger, the top finger. So Riley's showing you, she's going to pour in some water, about 3 fingers worth, into the bottle. And it helps to be warm water, but cool water will work too. It's just the warm water helps activate it a little bit quicker. All right, so take just a moment to make sure you've got your warm water, pour it into your container. And the next step, you're going to need your balloon. Now, for your balloon, you may have a different color, that's okay. But take your balloon and you want to stretch the mouth of the balloon a little bit, because you're going to need to put it over the top of the bottle. And this is where you may need a friend, right? You may need a friend to help you hold the bottle or to stretch the balloon over the top. You can also put the bottle between your knees and hold it really tightly and then stretch the balloon over the top. But that's what you need to do. So you're going to stretch that balloon. Okay, so what we're doing here is we have yeast, we have sugar, and we have warm water. Now the yeast was asleep, but we're waking it up with the warm water. We're giving it food. What do you think is going to happen? I want you to make a hypothesis or best guess, and you can put that into the chat if you like, about what is your hypothesis about what is going to happen to this bottle and to the balloon that's on the top of the bottle. And remember, a hypothesis, just that best scientific guess about what you think is going to happen to this fungus that we've just woken up. All right, anybody else? Okay, if you're ready to move on, then in the chat, let me know. All right, so making sure we have our yeast, our sugar, our bottle, and our warm water with the balloon over the top. Okay, while you're setting up, if you are still a little bit behind, that's absolutely okay. There are instructions in your booklets, and we're recording this video so you can watch it later. But right now, we're actually going to play an introduction video so that you can meet our scientists, Melissa and Aranga, and learn a little bit more about their research while our experiments sit and get going.
Hi, I'm really excited to be here with you today. My name is Dr. Melissa McCormick, and I run the Molecular Ecology Lab here at CERC. Now, molecular ecology just means that we use DNA to study a lot of different processes in ecology. And I use DNA especially for studying fungi. And the reason I do that is because a lot of fungi are very, very hard to see with our naked eye or with even a microscope, and it's very hard to identify them. So that's what I use the DNA to do.
Hi everyone, I'm Dr. Ranga, and I'm also very interested in learning about fungi. Specifically, I'm very curious to know how these plants find their fungal friends and also their enemies. So I'm working together with Melissa as a research fellow in her molecular ecology lab, and today you can learn some very cool stuff with us. So come on, let's go for a walk.
Today I'm recording this from one of my field sites, and I wanted to start out here in the woods to show you some of the places that I find fungi. But later we're going to go back to the lab, and I will be joining you live to discuss some of the fungi and some of the things that we study about fungi. So when I'm out here in the woods, I see a lot of the different but really important things that fungi do. And they do a lot of really important things. And one of the important things that they do is to feed plants. And for some plants, the fungi are so important that the plants can't grow there unless the right fungi are there.
Hey Melissa, what kind of plant is this?
This is a great example of a plant that needs fungi to grow. This is an orchid called a rattlesnake plantain. And the reason it's called a rattlesnake plantain is because when it sets fruit, the seed capsules look like the tail of a rattlesnake. And all orchids need fungi, and particular fungi, in order to grow. And those fungi are feeding on the leaves and the decomposing wood, and they're feeding it to the plant. And that's how the plants are able to grow. And all orchids, because they need specific fungi, their presence or where they grow is really driven by where the right fungi are. So in nature, every orchid depends on particular fungi to feed it, and many other plants do too. Those plants can't grow without their fungal helpers. This rattlesnake plantain can only be fed by one type of fungus, and then it transfers the nutrients, things like vitamins and minerals, to the orchid by decomposing the wood and leaves. And fungi can even communicate with each other and the plants that they live with. The fungi can send other fungi and plants signals through the soil, saying if it's dry, or if something is eating the plant, or it's under attack from something else. Why don't we take some of the soil and decomposing wood back to the lab and see if we can find fungi?
Oh, look at all the tree roots in this soil. All the trees need fungi to get nutrients too. And this decomposing wood has lots of nutrients in it. The fungi help them get nutrients and water and can even connect multiple trees together, letting them share nutrients.
Follow us back to the lab where we'll set up your experiment.
You can look at mushrooms up close with me.
Excellent. All right, everyone, welcome back. Thank you so much for joining us for our Science to Go. So we've just set up our experiments and we've been introduced to our scientists. We're now back in our laboratory, and I'm going to turn it over to Ms. Aranga, and she's going to help you with dissecting some mushroom gills, because we're talking about fungus and mushroom is a part of fungus. So Ms. Aranga, tell us a little bit more.
Hello, everyone. So now we are going to investigate a mushroom up close and personal. And I guess your mushrooms are moistened and ready to use. And I have my mushrooms moistened, and I have my tweezers, the napkin, and the index card ready to go. Now, can you remind me again what a mushroom is? Is it a plant? Is it an animal? Or is it some kind of fungi? You can put something in your chat box. So while you think of that, I'll take my mushroom outside.
And Miss Aranga, while you're doing that, everyone else can follow along too. So they can be getting their bag with the mushrooms that have been soaking in water, so they can follow along with you in this dissection.
Yep. All right. So I have my mushroom. Now you can take out your mushroom with your tweezers and keep it on the napkin.
If the tweezers are too hard to use, you can just use your fingers too. That's okay. But be careful not to spill the water. And when you're done with the baggie, just close it back up so the water doesn't spill everywhere.
Yeah. So now you can fold your napkin and tap it on the mushroom piece you have. And now you might see a brown patch.
Wait a minute, that brown looks kind of like the water. What's going on?
Yeah, let me focus it a little bit more. Okay, so yeah, so with all that water, you'll see a big brown patch here.
Are we looking at your screen, Miss Aranga? I don't see anything yet, but I'm excited to see what you're seeing.
Okay. I'm trying to share my screen.
Much better. Awesome.
Okay.
Hey, that's
Okay.
cool.
All right. Yeah. So if you keep the piece of mushroom on your napkin and tap on it with the napkin, you will see a big brown patch just like mine. And can you also see the water inside the bag has changed the color? You can think of why that happened, and you can answer that in the chat box as well. Why did it change the color? Okay, now we are going to look at this big brown patch a little bit closer. I know if you have your cell phone cameras or your tabs, you can try to look at it through the camera, but otherwise just eyeball it. And let's try to take out a little piece of gill out of this.
And Miss Aranga, when we're looking at this dehydrated mushroom, what are we looking at? Is this the stem? Is it the cap? Is it the underground part? What are we seeing?
Yeah, so let me tell that first. So maybe you have this diagram with you. So there are three main parts of the mushroom. So there's the cap, or which is like the umbrella-like part. The cap is the top of the mushroom. And then what we are looking at today is mostly the gills, this dark brown color part. The gills are very thin filaments. They are important because they produce spores. So what makes this big brown patch is the spores. So the spores inside these gills come out when they are rehydrated, and that is what's also changing the color of this water. These brown little spores are coming out of the gills. And then we also see the stem, which helps the mushroom to connect to the ground.
Okay, wait a minute, hang on now. You said it has gills. So does that mean that a mushroom's not a plant, but an animal or a fish with gills? Wait a minute.
No, Miss Karen. So this fungus they belong to their own kingdom. That is fungi. They are not animals. They are not plants. So they are coming under their own kingdom, fungi. So these spores are actually just like seeds of a fruit. So mushroom, we can call it a fruit or like a fruiting body, just like other plants make their fruits. These fungus make their fruiting body as a mushroom. So inside the mushroom, they have these spores, which then they can spread and make new fungus. Just like a seed can make a new plant, these fungal spores can make a new fungus. All right, so then you can take a little piece of gill with your tweezers. Maybe it might be a little difficult to take just one gill, but several gills might come out, but that's okay. Look at them carefully with your eyes. And
you can tear them with your fingers. And remember that your gill may look a little bit different, or your mushroom pieces may look a little different than Miss Aranga's. So don't worry about that. They're still all from the cap of the mushroom.
yes, while you look at them carefully, I will show you some actual spores that I took from the microscope. Can you all see that? So these, I took this microscope image from the same mushroom pieces. So these little oval shaped things are the actual spores which are coming out of the gills. So while you try to look at your gills.
So, Miss Aranga, does that mean I can see the spores with my eyes, or do I need a microscope, or both?
I think it's really difficult to see the spores with your naked eyes. But this big brown patch with little dots, that is made out of a lot of spores. But if you have a microscope, if you have the cell phone camera with a little lens, then you can try to look at the spores. Okay.
All right, that was really cool. Is there anything else you want to share with us about mushroom gills and spores?
So yeah, if you can take out, if you have a piece of the stem, you can take that and also look at that stem. The structure of the stem is a little bit different than the gills or the cap. So you can take out the piece of stem and look at that as well. So you might see a different structure with some shapes here in the stem. Yeah, you can also use this separated gills if you want to try growing your own mushrooms at home.
Oh,
I think you all
really
had
neat.
a nice, yeah. You can try that.
Let me ask you though, is this mushroom that you just rehydrated, is that the same thing I get in the grocery store?
Yeah, it's the same thing, but it's just dried.
Ah, okay. So those mushrooms we see in the grocery store are fungus, right? But wait a minute. You know, earlier I said yeast are fungus. How can they both be fungus?
Yeah, these are different species of fungus or different kinds of fungus. Sometimes they make a fruiting body or a mushroom, but some fungus, they do not make a mushroom.
Ah, okay. So I've heard that like mushrooms grow in the forest too. Can I eat those mushrooms?
Some of them, but you have to be very careful and you should know very well to identify them correctly to know whether it is edible or whether it is poisonous for you. So don't try that if you don't know.
Exactly. Always asking an adult. That's a great thing. So mushrooms are pretty neat. So they definitely are our friends for food. We know that because we can eat some of them. We see them in the grocery store. I even like them in my soup, which I think is kind of yummy. But mushrooms are also important for some other things. How about we ask Melissa what she thinks about fungus and how she looks at fungus? Hey, Melissa, you want to share with us what you do?
Sure. Hi, I'm in my lab right now, and I'm with some of my fungus samples. You can see some here. They're in these little tubes, and there's a whole bunch of them behind me in the cabinet. And we can talk about some of those during the question and answer session if people have questions later. But I wanted to first say, like Karen said, fungi come in a lot of different sizes, and they also do a lot of different things. And so the yeast that you have as part of your experiment, or that might be used to make bread, is very, very different than the mushrooms that you might get in the store or that you're dissected to be able to eat. And sometimes the fungi, like the yeast, are so small that you can't really see them with your naked eye. But other times they're bigger, like the mushroom you dissected or ones you might eat. And these, I'll see if I can show these to you here. Let's see. Oh, there's a lot of light. There we go. So there's one of the mushrooms that we grow, or fungi that we grow in the lab, and it's not making a mushroom, but it looks like that. That's the part of the fungus that we often don't see. So if you were to see a mushroom in the woods, what you wouldn't see was all the fungus that was growing underneath it, down in the soil. And so these fungi look like very, very different things. And this is just the ones that we're working with in the lab. And so there's a lot of things that are different. So sometimes fungi are a lot bigger, even than the mushrooms that you get in the store. And one thing that I like to ask people is, what do you think the biggest living creature is? And you can put some of the answers in the chat. And I see that Lexi says, I like mushrooms on my pizza, and I do too. I like mushrooms on a lot of things.
Well, while people are answering what folks think the biggest creature on Earth is, I got a question, Melissa. How does that fungus stick into that plate that you had? Because is that growing on something? What's going on?
Yeah, so these plates are plates that have like a gel, like Jello on them, and the fungus is growing in that surface, on top of that, and sort of sticking down into that gelatin surface. And so that's the way that they're growing right now. And so if they were in soil, they'd be down in the soil, down where you can't see them.
So is that like the stuff that grows on the bread that molds in my fridge?
It's not very closely related, but yes, it's doing the same thing. So I see some guesses in the chat. I see somebody thought there was a blue whale. Somebody thought an elephant. Some people have suggested a giant redwood as a possibility. But it turns out that the biggest living creature is actually a fungus. There's a honey fungus that's growing in a forest in Oregon, where a single fungus is almost 2.5 miles across. And scientists think it's probably over 8,000 years old. Now, if you walked 2.5 miles, it would probably take you over an hour. Now, if you looked at the ground in this forest, you'd probably see occasional mushrooms popping up here and there. It wouldn't be like the whole thing was a giant sheet of fungus. But if you looked at under the ground, all those mushrooms would be connected to each other down in the soil with all of these threads of fungus all growing together. And you can think of the mushrooms that you see above ground as kind of like the flowers on a plant. So if you look at the flowers on a plant, well, you wouldn't think that each flower was a separate plant. No, they're just separate flowers on a single plant. And the fungus is sort of the same way. Most of the fungus is actually below ground. It's in the soil, and it's growing in the soil and in the decomposing wood, and it's not the part that you see. And so mushrooms are for fungi, kind of like that. They're the fruiting structure. They are what produces the spores that make the new mushrooms. That make the new fungi. And so I study what different fungi do to help plants grow. And I mentioned that a little bit in the video, but I try to understand how fungi feed plants and how the fungi that grow in an area can determine what plants grow there. Only some fungi can feed plants. Other fungi don't feed plants, but they still do important things. Some of them decompose or break down wood and fallen leaves into food that other plants and animals can eat. Now, all fungi have a job in the forest. A lot of them help plants, but not all. Almost all plants need some fungi in order to be able to grow well. They can't get the food that they need and the water that they need without the help of fungi. And some plants need particular fungi to be able to grow, like orchids, like the ones that we saw in the video in the woods. That's one group of plants that needs specific fungi. Just the right fungus has to be there in order for the orchid to be able to grow there. So if you don't have the right fungus, you don't get the orchid. And other plants maybe don't need just one fungus. Maybe they can use 2 or 3 or even 15, but still that's a very, very small number of fungi that they can possibly use in order to grow. And those fungi often have to be there in order for the plants to grow there. Now,
Okay, wait a minute. Hang on. Let me ask you a question, because you said that little plant you saw in the forest was an orchid. I thought orchids all look like the things in the grocery stores. Do they have fungus, and are those orchids?
So the orchids in the grocery store probably don't have fungus. And the only reason they don't have fungus is because somebody grew them in the lab under very special conditions that fed the orchid just exactly the food that it needed. Now, if that orchid were growing in nature, it would need to have a fungus with it, and it would. But when we do things in the lab, maybe that doesn't always happen. We have some orchids in the lab here that we can grow without a fungus, but for them to be able to survive, if we planted them outside, then they would need to have an association with a fungus in order to be able to get the food that they need. And to be able to grow from the seed.
That's cool. Thank you.
And so Uranga is working to understand how orchids and fungi work together, how they communicate with each other so that they can grow together. How does this orchid know whether this is the right fungus for it or not? And so I want to know how fungi and plants work together in order to allow the plants to grow in different places. I also want to know how we can work with fungi to grow more native orchids. And that's one of the things I really enjoy working on. Now, a lot of orchids are very rare, and to be sure that they can survive or to grow enough to put back into the wild, we need to be able to be sure that we save the fungi that they need too. We can't just gather up a lot of orchid seeds without having the right fungus. If you don't have the right fungus, those seeds can't ever grow into plants. And so you have to have both in order for it to work. Now, the fungi feed the plants, and not just orchids, but all plants, almost all plants, by attaching to their roots underground. And they form this big network, kind of like the internet, where plants can talk and communicate. And when you see a tree, for example, you only see what's above ground, but there's more tree below ground that you don't see than there is above ground. So there's an awful lot going on underneath your feet that you just don't even think about or see. And plants can send signals to other plants, asking them maybe for nutrients or letting other plants know that they're under attack by insects. And so they better ramp up their defenses so they don't get those insects eating them too. And a lot of this they do through their fungi. So when I learned that fungi could even allow plants to talk to each other, to some degree, of course, and to grow in places that they couldn't grow alone, I realized how much of learning why plants grow where they do depended on solving the puzzles of why particular fungi grow where they do. And we know a whole lot less about why fungi grow where they do than we do about plants. And part of that is just because most of the fungus we can't see, it's below ground. It's a lot easier to go and look and see the plants than it is to see the fungus that's in the soil. And so it's taken a lot of development of science, in particular working with DNA. And one of the other videos in this Science to Go group is going to talk to you about DNA. But I work a lot with DNA also. But these puzzles of trying to figure out why fungi grow where they do and how that means how that determines where plants can grow where they do is like a super puzzle for me. And I love puzzles. And I don't know how many of you really like puzzles or mysteries, but to me, that's what doing research is about. It's about solving the puzzles and solving the mysteries. And I get to decide what puzzles and what mysteries I want to try to solve and why they're interesting. And so doing research for me is sort of like the ultimate puzzle. And it's what I work really, really hard to understand. And so if it's something that you think you might be interested in, you can do research in a lot of different ways. And there are also opportunities to interact with other groups as well doing research. And so you might think about it if you really like puzzles and mysteries.
Oh, wow, that's really neat. Well, question for you, because I've always wondered this. If I go out on a hike right now in the winter time, can I see fungus?
You can, although most of the fungus that you will see will be, some fungi produce structures, mushrooms that can dry out in place. And just like the dried out mushroom that you have that you received in your kit, you can see dried mushrooms that are on the sides of trees or on decomposing logs or on the soil. And most of those will be fungi that produced their mushrooms in the fall, and they've now dried out somewhat, but they're still there. And if you get a warm spell in the spring, sometimes the mushrooms will start to pop up during the warm spells as well, particularly if it's warm and wet.
Oh, okay. And then what is that stuff though? Like I see it on, and you said decomposing or like breaking down logs. What is that white stuff that I see? Is that fungus?
Usually, yes, it is fungus.
Okay.
So that's
cool.
usually the strands of fungus. It's not the mushrooms, it's the strands of fungus growing together and growing into the bits of wood that then they will break down to make food for other plants and animals.
Ah, okay. All right. So that's still a fungus, but it's sort of like the stuff in your plates, right? That you showed us earlier. Is that the same type of thing?
It is. It is. And so what
Okay.
you see on the plates is sort of the strands of fungus. And if I looked at it under a microscope, I would see like a net of fungus on the surface. And somebody asked if lichens were the same. And lichens are a plant and a fungus living together, but the plant is the microscopic part, the little part, and the fungus is the bigger part. So what you see in a lichen is usually algae, so little single-celled plants living inside of fungal tissue. And so that's what you see when you see a lichen.
You know how I remember that? Allison Algae took, Allison Algae and Freddie Fungus took a lichen to each other. So little phrases always help me remember things in biology, right? So, hey, we all have our things. So very cool. So if there's a lichen and there's like algae, that means that the algae is making the food like a plant, right? And the fungus is like the body. Is that what's going on?
So, well, the algae are making the carbon food. They're making the sugars that the fungus needs, but the fungus is getting access to minerals and vitamins and stuff in the soil or on the rocks that it's growing on. And they're feeding that also to the algae as well as to the fungus. So they're doing different things and they're working together so that they can both grow.
Excellent. So this kind of all goes back to that idea that fungus is our friend in our food, what we eat, but also in the forest because they are helping break down dead stuff and they're helping plants and trees uptake nutrients and the things that they need to survive. All sorts of neat things come out of fungus. That's pretty neat. So, hey, you know what? We still have an experiment running. So I thought this would be a great time to take a look and see what's happening with our experiment right now. Miss Riley, do you have yours running where you can show us what you've got? How about you all in the classroom? Do you have your experiment going? What do you observe? Can you put your results in the chat and let us know what you see? And then did this support or not support your hypothesis? Hey, check that out. This is pretty cool. So it looks like Riley's has gotten, the balloon's gotten pretty big. So we see that the top is filled with something. And then the bottom part looks like it's kind of filled up. Does anyone know what's going on with that? What do you notice? And if you look at it, the yeast is taking up more of the bottle. Do you observe any air or any little bubbles in there? What's happening? Does your experiment match what Riley's looks like? Like mine is actually even more full. I used really warm water. So what's happening here? Think about yeast, all right? It's not a plant, but it still eats and breathes because it's a living thing, right? So what do you think is happening with that sugar in there? Well, I'll tell you this, because it's a creature and it needs to eat, well, we've just fed it, right? So that yeast is consuming or eating the sugar, just like when you make bread, you put a little bit of sugar, you might put some flour into your bread. We've fed the yeast, we've activated it or woken it up with some water. And then what it's doing is it is exuding or releasing carbon dioxide. So it's taking in oxygen and releasing carbon dioxide, very similar to the way that we breathe and just similar to the way that we eat too. So this is the thing. Have you ever seen holes in bread that you eat? Those holes are left behind by gas bubbles, just like the gas bubbles that you see inside your bottle, right? And those gas bubbles are released by the yeast as they're eating the sugar from the bread and the carbohydrates that they are releasing carbon dioxide and leaving behind those holes. And so yeast is used to make bread be light and airy and to make it rise. What do you think would happen if we had bread that we made without yeast? What would happen? Any ideas? Would it rise or would it be flat like a cracker? Any ideas? Well, if you said flat like a cracker, you'd be right. We need that yeast to make the carbon dioxide and those bubbles to make the bread rise. So it's important. And yeast is also important in other foods for fermentation or for breaking down that food to create things like soy sauce or to create things like pickled cabbage or to create things like some of the drinks that we drink that are fermented drinks, all right? So with that being said, we know that there's carbon dioxide in the bottle. We know it's filling with air because there's something being captured in here. Here's my question for you. What do you think would happen if we let this balloon and bottle sit all day long? Would the balloon continue to blow up and eventually explode? Would all of the oxygen eventually get used up and the balloon shrink down? Would it stay just the same? What do you think might happen? Now, I'm not going to tell you the answer to that because I'm going to let you find out in your classrooms or at home. And if you want to, you can also do this same experiment with using different amounts of sugar, using different amounts of yeast, or even adding a little bit of flour. So all of these things can be done with your experiment. Now, when you clean up your experiment, something that we can do is we can actually, very carefully, don't do this yet, we can take this balloon off and pinch it off. And if you hold it right side up, and I'll show you, don't do this. If you hold it, you can actually let the mouth of the balloon go very slowly. And you can hear the gas being released. You can also smell what smells like fermentation. And that fermentation is exactly what's happening. The yeast is eating the sugar and releasing the carbon dioxide gas. And it smells really funky, right? But that fermentation is super important for releasing the bubbles from the food. Now, when you go to clean up your bottles, it's really important that you hold it over a sink or that you hold it over something that can get messy because some of the liquid may come out of the balloon and some of the liquid may overflow the bottle. So make sure that when you get to the point where you're ready to take that balloon off and you're ready to let the gas out of that balloon, that you do it over a sink or something that can get messy and kind of gross. All right. Well, this has been super fun learning about different types of fungus. We've learned about mushrooms, the type that we eat that are friends for our food and for our homes, the things that we like to consume. We've learned about another type of fungus, which is the yeast that we use in our bread and in some of our foods, things like soy sauce. But this is a thing. Fungus is all around us. And the next time you go outside, you go on a hike, or you even are walking around where there may be wood or leaves, you can lift those up and look for the little white filaments that are fungus, or look for old dried up mushrooms and see if you can find them. You might even be able to find them if you open up the door to your fridge. Now, remember, don't eat the mushrooms and fungus you find in the forest, right? Only the safe ones from the grocery store. And the ones that adults say are safe to eat. So fungus are our friends. They're in food and they're in forests, and they're also used for antibiotics. They're super important. Now, before we finish up, we had some really wonderful questions in the chat, and I'd like to go back to Melissa and Aranga to take a few more questions to answer some of those things that you wanted to know a little bit more about. And with that, I'm going to turn it over to Miss Emily to read off any of the questions that they have put into the chat or other places. So go ahead, Miss Emily.
Hi, everybody. Thank you so much for all your awesome questions. And Melissa and Aranga, I have a couple of questions for you. Specifically, how, as you as a scientist, how would you classify fungi? I know we talked about how fungi is not a plant and not animals. What makes us separate the two or the three?
So we classify fungi, so we separate fungi from plants or animals because of the types of, the way that their cells are structured. So at the very, very, very small level, all of the pieces that make up a fungus, they function very differently than the cells of a plant do. And most plants they make their own food through photosynthesis. Now they still have to get things from the soil. They still have to get vitamins and minerals and all sorts of nutrients like that. But the carbon that they get, the sugars that they make from photosynthesis, from using sunlight, that's something that no fungi can do. So fungi always have to eat other things in order to grow, just like animals do. Animals also have to eat other things. But the cells of fungi are very different from the cells of animals. And so that's sort of where we make that first, how we know fungi are very different from plants and animals. But we classify the fungi usually based on the type of mushroom that they make and what the structure of that mushroom is. So you might see mushrooms that look very, very different. You might see mushrooms that instead of having gills, have little pores, little holes on the underside of the mushroom cap. And they have spores up inside those little pores, but they're not on gills. That's a different type of fungus. And that's one of the ways that we would classify the fungi, is based on the type of mushroom that they make. But not all fungi make mushrooms. And the ones, like some of the ones that I've shown you that we work with in the lab, they don't make mushrooms. And that's one of the reasons we know so little about them, because they don't ever really make anything that we can look at above ground and say, oh, that's a chanterelle, or that's a morel mushroom. You don't see anything. And so we know very little about where they grow and what they do, but we know that they have to be there for different orchids to grow. And so that's something that the ways of classifying fungi are starting to change now. We can use a little piece of DNA sequence, a little piece of the instructions that are used by the fungus for making fungus in order to identify them. And so that's starting to change how we identify different fungi.
Oh, that's really cool. I couldn't even imagine walking through the forest and knowing that there's so much fungi in the ground underneath. How do you find them other than the DNA? Do you just dig in the dirt, or how are you able to study them if they don't make fruiting bodies like mushrooms?
So yes, that's a good question. And if we take an amount of soil about the size of a lima bean, so just a little bit, or like a little teaspoon, that fungus probably, that soil probably has about 150 different species of fungus in it. And if we went this far away from it and took another little piece of soil, that amount, it would only share 1/4 of the fungus species with that other sample that was just that far apart. And so there's a huge diversity of fungi. There's a lot of different fungi that live in the soil. And in order to figure out what fungi are there, we use DNA to do that. Now, what they used to have to do was they would take that soil and they'd grind it all up and they'd thin it out with lots and lots and lots of water and then spread it out on all kinds of different nutrient augers. So like the gel that was in the plates that our fungus is growing on, and they'd see what grew. Of course, some fungi don't grow when you do that. And so that's one of the reasons that we use DNA to do that. And so we can take that little piece of soil and we can see how many different sequences of a little particular piece of DNA that we use as sort of, they call it a barcoding region. So it's a region that, just like a barcode in the store, allows the scanner to tell what fruit you have or what box of crackers you have and how much it costs. Well, we can use that little piece of DNA, the sequence from that little piece of DNA, just like a barcode, to know what fungus we found in the soil. And so that allows us to do that. We can look at it with a microscope, but you can't always tell them apart just by looking at them.
And how did you become interested in studying fungi and orchids? And how, if there was someone interested, could they start studying that sort of thing?
So I grew up with plants. My mom was a master gardener, and so we always had plants growing in the yard, in the house. I spent a lot of time outside hiking as a kid and seeing all of the different plants that grew different places. And I didn't think a whole lot about the fungi. And it wasn't until I was in, I want to say, college, that I realized that behind all of those plants that I saw and found so interesting was actually a bunch of different fungi that were enabling plants to grow where they did. And so that was just sort of a mystery that just really hooked me, just like a really good mystery book. If you're reading it, you get hooked in it, you just can't put it down. Well, for me, that's what understanding the relationship between fungi and plants was. And orchids are sort of, they really, really, really need their fungi, even more so than most plants. And they actually won't even grow from seeds if they don't have the right fungi. And so they're just sort of an extreme example. And of course, orchids are pretty, which helps. And they use a lot of different fungi that we know very little about. So there's an awful lot that's not known for me to be just able to try to figure out. And if somebody wanted to try to learn about fungi, I think there are a lot of things that you can do yourself. There are a lot of activities that you can find online. And when you go to college, of course, then there are opportunities to study fungi and lots of school beyond that. So you have to really like your mysteries. But there are opportunities, and I think Miss Karen might have some ideas, because she works with education at CERC, and so that might be a good place to check as well.
Oh, you're totally right. We'll definitely be doing some programs coming soon as the weather gets a little bit warmer. So thank you so much. I did have another question that came up in the Q&A, and Aranga, if you want to answer this, and Melissa, I think both of you, is someone asked, did you have challenges as a woman scientist? And I see you're in the research labs. You've been successful for a long time. What has made you successful in the field?
I think I came from a very distant place in the world. I came originally from Sri Lanka, which is a tropical country, and that is one reason that I'm very fascinated about orchids. I also grew up with looking at lots of tropical orchids, the diversity in them, and I was fascinated by the diversity, and I wanted to learn about the orchids, and so that's what brought me here to the United States. So that itself was a challenge for me as a young graduate to come here. And but still, I mean, if you are passionate about something, if you really want to learn something, I think you will make the background by yourself if you have that passion. No matter what, if you're passionate about it, you will do it. So so when I got here also, I had to solve problems by myself. And as a girl, as a woman scientist, it is true that you get lots of challenges. I mean, for me, even driving on a highway was a challenge for me. So even that, you might say it's a very simple thing, but for me, even that was a challenge. So if you don't see that as a challenge, you are way ahead of me. So most of you can do that. So don't worry if you're passionate of learning things. So just follow your goal and you'll get there.
And how about you, Melissa?
So for me, I think as a woman scientist, it can be challenging when you work with, I work with mostly men, less so now, very much less so now than it used to be when I first got started. And that was 20 some years ago. So things are changing and they're getting much better. As a woman scientist, sometimes there are men who don't treat you like, they don't recognize your experience and contributions to the same degree. And I think that is very much changing. And I know that I have 2 daughters and they are both teenagers, and both of them said, wait, what? They did that? I don't understand. Why would somebody do that? Why would somebody ever think that women were less capable than men? And I talked to some of the women scientists who are much older than me, and the stories that they can tell are far worse than anything I experienced. So I would say, absolutely, a woman can do it. Any woman can do it just as well as a man can. And that the diversity that you get from learning from different people, both men and women, people from a variety of different backgrounds, all of that contributes an awful lot to understanding, to learning new things and to functioning as a better scientific organization. And Karen asked what she thought, or somebody asked what they thought contributed to my success as a scientist. And I would say that it's creativity, and creativity requires that you think about things differently, and you think about things from different perspectives, and you consider all kinds of different perspectives that come from people from different backgrounds. And that's what's so important to making big discoveries in science. And just a little plug, if you are currently in the DC area, there is an exhibit at the Kogod Courtyard, which is one of the Smithsonian museums, called that is about the hidden contributions of women to orchid science. And I am one of the featured scientists in that. So along with a lot of very, very accomplished women scientists and artists. And so if you have a chance to go take a look at it, that would be a great opportunity to see some of the really cool things that women have contributed.
Oh my gosh, that's amazing. Thank you so much. We're running out of time, and I do have one last thing we need to do. But Melissa, Aranga, thank you so much for joining us today. We've had a great time learning about fungus and the importance to our food and our ecosystems and forests and homes. And I really look forward to seeing where your research takes you. Thank you again. And with that, for those of you who are students and in the classroom, that's the end of our program. But if you're a teacher or educator, we have a last minute request so that we can continue funding these activities and programs and giving out our science kits for free. So Lexi, do you want to tell them what we have?
Yes, I do. Hello. Thank you again so much for attending today. Thank you so much, Melissa and Aranga. So once again, students, if you could log off, this is just directly to the educators. As Karen said, partly for our funding and also for my master's degree, I am going to be running surveys where I'm collecting some information about your opinions and experiences in these professional development workshops. So not just the professional development workshops, but also these live Science to Go events. So in the chat, I will post right now a link to this super super brief survey, and I would really appreciate it if you could take it. It will literally take like 3 minutes of your time. Super easy. And yes, please remember, this is only for teachers. Students, thank you for watching and participating, but teachers, please only do this. Thank you.
All right. Thanks, Lexi. Thanks everyone for joining us. Have a wonderful rest of your week. And hey, keep doing science. We hope that you will be one of our future scientists, and maybe someday you can join us as an intern or researcher at the Smithsonian Environmental Research Center. Thanks for joining.